fluorescent intensity and concentration of the analyte. The relationship between the fluorescent intensity of the solution to the concentration of that solution is a linear relationship passing through the origin. So F, which is the fluorescence or fluorescence intensity, is directly proportional to the concentration. We are introducing A where it is the proportionality constant. So fluorescence intensity depends linearly on concentration. Please note that there is no constant. Deviation occurs at high concentrations. Now, as there was a limitation to Beer-Lambert's law, there will be limitations for this particular law also of fluorescence. Uh, so deviation will be similar to those what we observed for UV spectroscopy. First will be the self-absorption at high concentration. Neighboring molecules absorb emitted photons from other molecules. So one molecule is emitting the photon, but that emitted photon is absorbed by the other molecule within the same solvent. So overall, from the solution, no photon got escaped, although there was transition taking place from, say, excited singlet to ground state. So although there was transition, there was photon emitted, but from the solution as a overall, no photon was emitted. So detector detected nothing. So it is less fluorescence than what is actually expected. So it happens if there is overlap between excitation and emission spectra. It can happen that a compound emits a large spectrum of photon when it is coming down to the ground state. If that is the case, if such is the case, then the excited emitted photon may get absorbed by the other molecule. Now, second reason is quenching. Now, for quenching, there are multiple reasons, but this reason is concentration related. Collision of excited state molecule with other excited state molecules and the radiationless deactivation may occur. So there is mutual exchange of energy and that exchange is radiationless. Now, the first point was related to concentration. Next is deviation from high excitation light intensity. When we say high excitation light intensity, simply saying that the electromagnetic radiation which is reaching to the molecule in your analyte has very large intensity. When we say large intensity, we mean to say it has got high energy associated with it. If we provide sufficiently high energy to the molecule, what will happen to the molecule? It will break down. So this is called as photo bleaching. Excited state molecule absorb another photon and is destroyed. Destroyed excited state molecule is not able to emit fluorescent photon. So that degraded product cannot emit photon because the energy has been utilized in breaking down of the molecule. Now, in general, what is bleaching? Any loss in the color is called as bleaching. Photo bleaching, loss in that absorption and emission because of energy provided by the photon will be called as photo bleaching. Let us talk more about those excitation and emission spectra. Okay, the deviation is over now. There were two aspects of it, one related to concentration and one related to intensity of the incident electromagnetic radiation. Okay, so there will be always two part to the spectra of the molecules showing fluorescence. One will be absorption spectra, which is also called as excitation spectra, and the other will be emission spectra. See, any molecule, if you consider just the absorbance in UV spectroscopy region, it was not that only one wavelength is absorbed by the molecule. Molecule absorbs a set of wavelengths, band of spectra the molecule absorbs. If band of spectra is absorbed by the molecule, emit the energy which is less than what is absorbed. So it is going to emit the longer wavelength. It will not emit single longer wavelength. It will emit the set of longer wavelengths that molecule. So molecule will have two spectra. excitation spectra and emission spectra. See what is excitation spectra is. Emission wavelength is fixed. Excitation wavelength is scanned. So how to obtain this spectrum? We are going to fix the wavelength that is measured by the detector, emission wavelength that is the measured by the detector, by adjusting that wavelength selector on that side. But the excitation wavelength will be scanned. So monochromator or filter selected to allow only one wavelength of fluorescent light to pass through to the detector. Excitation wavelength is varied. At each excitation wavelength increment, fluon at the fixed emission of wavelength are collected. The emission intensity, that is, the number of fluorescent photons collected at each wavelength increment, varies as the excitation wavelength comes closer to or goes further from wavelength of maximum absorption. This is why an excitation spectra looks like an absorption spectra. So in fluorescence spectroscopy, there will be source of electromagnetic radiation. Then there will be a monochromator. This is first monochromator, which will control the excitation wavelength. From this first monochromator, only one wavelength, electromagnetic radiation of only one wavelength, will go to the sample. This is your sample. Okay. From the sample, the emission spectra will go to monochromator 2. Now, careful listen why what did I say? I said excitation spectra converted to single wavelength. Then there is emission spectra coming over here. It will be converted into single wavelength, and then it will be detected by the detector. Then it will be amplified. Then you will get the reading. Now, what I'm trying to say here is overall what is happening in the process. So what is detector is measuring? Detector is measuring this wavelength selected by this detector. It is getting measured by the detector. This intensity of wavelength that is coming from over here will depend on how much energy was absorbed by the sample. How much energy sample is absorbing. Sample is not absorbing at all the wavelengths equally. It will have one wavelength at which it is getting excited at maximum. So what we are going to do when we are observing this spectra of excitation wavelength, we are going to keep this wavelength fixed, and we are going to change the excitation wavelength. So when excitation wavelength is being changed, we are actually measuring the emission wavelength itself. We are measuring the intensity of emission wavelength. This intensity of measured is of emitted light, a fixed wavelength. But this wavelength that is being changed is excitation. This is emission and this is excitation. In absorption, what we did, we changed the wavelength and we measured the same wavelength. But here we are changing a different wavelength and we are measuring intensity of a emitted wavelength. So as excitation wavelength changes, there is no change in the intensity, or it is very less intensity of the emission wavelength. But there will be certain excitation wavelength at which intensity of the emitted light of that particular wavelength will go on increasing, and then again it will go on decreasing. So we will conclude that in this region the molecule is absorbing maximum electromagnetic radiation, and therefore it is also emitting that particular wavelength that we have selected at different intensity. See, excitation wavelength is fixed, So we are going to keep the wavelength fixed by which we are exciting the molecule. Probably we will select the same wavelength which excited that molecule the most. Okay, so we are going to keep that wavelength constant, and we are going to change the wavelength that is selected by the detector, that is detected by the detector, that is emission wavelength. Monochromator or filter is selected to allow only one wavelength of excitation light to pass into the sample. The emission wavelength is varied. We are going to use monochromator to change that wavelength, and we are going to plot the spectra. So fluorescent photons are collected at each incremental emission wavelength. So on every wavelength we are going to check their intensity. The emission intensity, that is number of fluorescent photons collected at each wavelength increment, varies as the emission wavelength changed. The emission intensity, that is the number of fluorescent photons collected at each wavelength increment, varies as the emission wavelength is changed. Spectrum shows at what wavelength the fluorescent intensity is maximum given excitation wavelength. So if we consider that both the things we are measuring, this part of the spectra, left hand side part, is excitation spectra. And from excitation spectra, suppose we selected 250 nanometer wavelength and plotted the emission spectra. So you will get this emission spectra. So this is how those excitation and emission spectra looks like. Observe the diagrams, you will get some more details. This is the spectra is drawn for 1 ppm solution of anthracene in alcohol. Can you make some observation regarding these spectra, which is very obvious from their shapes? Do you think they are mirror images of each other?
Yes, sir.
They are not exactly same, they are mirror images of each other. We need to come to the quenching now. The quenching is decrease in the intensity, the fluorescence. There are certain types of quenching. One is called as dynamic quenching. The other quenching is resulting from dissolved oxygen, and there is also something called as static quenching. So let us see all of them one by one. In dynamic quenching, radiative energy transfer between the excited state and quenching agent species occurs. So for dynamic quenching, we will require one agent which will act as quenching agent. So energy will be transferred from the excited molecule to this quenching agent instead of released as a photon. The requirements for successful dynamic quenching are that oxygen species concentration must be high so that there is higher possibility of the collision between two species. So the concentration of that quenching agent should be sufficiently high that the quenching agent collides with the excited molecule. The excited molecule being the first species and the quenching molecule being the second species. Temperature and quenching agent viscosity play a role on the rate of dynamic quenching. Now, what is your guess? As temperature increases, quenching will increase or quenching will decrease. If the temperature increases, movement of the molecule will also increase. If temperature increases, the quenching will also increase. What do you think about viscosity? What will happen if viscosity increases? What will happen? Dynamic quenching reduces fluorescence quantum yield and fluorescence lifetime both. The species will fluoresce for short period of time and overall quenching in the solution will also decrease. Next point is more of a reason. So it is dissolved oxygen. Dissolved oxygen in solution increases the intensity of fluorescence by photochemically inducing oxidation of fluorescing species. So basically what is happening is it is resulting in a chemical reaction But this is only one aspect of the scenario. There is other aspect also. Quenching results from paramagnetic properties of molecular oxygen that promotes inter-system crossing and converts the excited molecules to triplet state. Now, this paramagnetic properties. I hope you know the magnetic properties. There is diamagnetic, there is paramagnetic, and there is non-magnetic. So basically, the paramagnetic properties helps that molecular oxygen to promote the intersystem crossing in the adjacent species. So paramagnetic species and dissolved oxygen tend to quench the fluorescence and quench the triplet state. Static quenching. This is the third part. Static quenching occurs when the quencher and the ground state fluorophore forms a dark complex. It is basically a quencher is getting associated with the fluorophore of the compound. When it is getting associated with fluorophore compound, there is no photon that is emitted from that complex. The energy is absorbed or distributed throughout that complex. Fluorescence is usually observed from unbound fluorophore. The fluorophore should be free, then only fluorescence is observed. Static quenching can be differentiated from dynamic quenching in that lifetime is not affected in static quenching. So the lifetime remains the same, only the intensity decreases of the solution overall. For long range quenching, energy transfer occurs without collision between molecules, but dipole dipole coupling occurs between excited fluorophore and quencher. When we say long range quenching, in this case, the quencher is not in directly contact with the analyte, or I should say the fluorophore. It is not associated, but it is in the proximity. Okay, that's why it is called as long range quenching. Now this occurs because of dipole dipole moment. It affects how electrons behave. Now, next part of the chapter is instrumentation. We need to study sources, filters or monochromators that are used. There will be two types of filters and monochromators that can be used, one for excitation and other for emission. And we will have detector. This is the block diagram. Go through it. So this is the optical diagram. Please note that detector, that is the photomultiplier tube, is kept at 90 degrees of the source from the sample. This is ray diagram. This is the source over here. From the source, electromagnetic radiation are collimated into the excitation monochromator. From excitation monochromator, we are going to select one wavelength. That single wavelength is made to fall on the sample. Okay, there is a beam splitter. The beam splitter focuses the original excitation monochromator into the detector compartment. Basically, what happens is it is you can say diverted into that compartment. There is one, you can say, absorbance compensating cell. Basically, it is going to absorb the intensity of light and reducing that light. And then there will be one white reflector kept over here. So there will be a sample compartment over here. From sample compartment, some wavelength will be absorbed by sample and the fluorescence radiation will be emitted by the sample, which will be again enter into emission monochromator. Emission monochromator will select the wavelength that is getting detected by the detector. Now, whatever reading is observed over here and whatever reading observed over here in a reference detector, both will be relayed to the detector system. The detector system will subtract a reading at the reference from the sample and give you the reading for the actual fluorescence. So this is the role of this setup. It is acting as a reference. The application involves determination of inorganic substance. Now, inorganic substances, you can say the of this is the substance should either possess the fluorescence by itself, or it may happen that the inorganic substance is responsible for the quenching. In both the cases, the substance may be detected as the fluorescent species, or it may be determined as the quenching species. So, determination of ruthenium ion in presence of other platinum metals. Now, ruthenium ion and the platinum metals here both are, you can say, the large species. Platinum metal acts as you can say quencher. Okay, so ruthenium ion is getting quenched. So decrease in the intensity of ruthenium ion is indication of presence of the platinum metal. So standard graph is plotted, and then the fluorescence of ruthenium ion is determined uh, by the by the fluorescence of the unknown. Determination of aluminum 3 alloys. Then aluminum 3 is a quencher. Determination of boron in steel by complex formed with steel is uh, a quencher. Now, in nuclear research, now the electromagnetic radiation coming from the nuclear material is responsible for causing fluorescence. So that field determination of uranium salts can be done by fluorescence substance. Mini fluorescent indicator mainly used in acid-base titration, which gives you the end point in certain pH. We have seen that pH is, if you change the pH, if you change the circumstances, the fluorescence, the fluorophore will change. So appearance or disappearance of the fluorescent color is indicator of the end point. Then organic analysis, qualitative and quantitative analysis of organic aromatic compounds present in cigarette smoke, air pollutants, automobile exhaust, and obviously it is also related to pharmaceutical analysis. So there are certain compounds which gives fluorescence. One such example is hydrocortisone. When it is reacted with 75% sulfuric acid in ethanol, a complex is formed which is post oxidation complex. It has got excitation index of 260 and fluorescence wavelength of 520. Then there is application in liquid chromatography. These fluorescent detectors are used as detector in liquid chromatography. Fluorescence is an important method of determination of compounds as they appear at the end of chromatogram and capillary electrophoresis column. Then determination of vitamin B1 and B12, both of which are fluorescent species.